Hello everyone. So this presentation will be handling the extractable study design and challenges that are associated with large volume parenterals. So in this presentation we will go over some definitions. We will look at the regulatory guidelines that are out there for parenteral applications. We will look at the potential sources for extractables and leachables in flexible bag systems. The challenges that are associated with large volume parenterals applications will be looked at, and we will look into material assessment and packaging assessment that can both be used as a setup for extractable studies. So let's look first at some definitions. A large volume parenteral application is a unit that is administered in one single dose and that contains a volume of over 100 milliliters that is administered by parenteral route. So the large volume parenteral applications are in that way quite different from small volume parenteral applications, since they contain a large volume that is administered at once in one single dose, where small volume containers contain smaller volumes of only a few milliliters that might be administered in multiple number of doses. And this difference will have a big impact on the challenges that are associated with those containers. So let's look at the definition of extractables. An extractable study is focused on the packaging materials, both primary and secondary packaging materials, that are submerged in an extraction solvent and extracted under laboratory conditions. So the setup of an extractable study can be quite different depending on the goal of the study. This can be material characterization and material selection, and or preparing for a leachable study. Of course, it's also important in the design of an extractable study that the requirements as being put forward by the authorities, the FDA and the EMA, are taken into account. A leachable study, on the other hand, focuses on the drug product after it has been in contact with the packaging system and the packaging materials under intended use conditions. So the impurities that are present in the drug product as a result of the contact with the packaging can then be evaluated for both the toxicity towards the patient and the impact on drug potency. So if we look at the differences between extractables and leachable studies, an extractable study is focused on a pharmaceutical container, which is subjected to worst-case conditions in order to identify the compounds that can potentially come out of the container. The analytical techniques that are used for these studies are screening analysis, and these analytical techniques are designed to screen for a very wide range of potential compounds that can migrate from these materials. And my colleague Barto Tri will explain later on in his presentation more how such a screening approach works. On the other hand, the leachable study is focused on the drug product that is in contact with the packaging material under normal conditions in order to quantify the compounds that do come out in reality. And for that purpose, both targeted analytical techniques are used, as well as screening analysis to account for unexpected leachables that might be present. So an extractable study is an important preparation or preparative study to go into a leachable study. And in order to go from an extractable study to a leachable study, the extractable study has the purpose to identify the compounds that need to be quantified in the leachable study. And the selection of those compounds is based on toxicological principles and safety thresholds. And this principle will be very important in large volume parenteral applications. We will further discuss that later on in the presentation, and this will also be explained further in the presentation of my colleague Peter van Wouwen on leachable studies. So let's look at the regulatory guidelines then. In 1999, the FDA published the first guideline on container closure systems for packaging human drugs and biologics. This is already quite some time ago, and to be honest, this guideline is already a bit outdated. In 2005, the European Medicines Agency also came out with a guideline on plastic immediate packaging materials, and that guideline contained a decision tree on what information needs to be provided towards the authorities for different dosage forms. But most recently, the US Pharmacopoeia published 3 important chapters, 1661, 1663 and 1664, on extractables and leachables testing, and those guidelines are actually in line with the most recent expectations of the authorities. So if we zoom in on the USP 1663 monograph, so these monographs are numbered above 1,000. That means that these are informal monographs, and you will also see that the content of this monograph is still open for interpretation, but it provides guidance on how to set up extractable studies and what important factors need to be taken into account. This monograph also contains a classification scheme for different products in regards with their degree of concern towards extractables and leachables studies. And this classification scheme was copied from the original FDA guideline. And for parenteral applications, we see that the degree that is associated with route of administration is the highest, while the likelihood of interaction between the packaging material and the dosage form is medium. So this results in a quite high risk for extractables and leachables associated with large volume parenteral applications. So besides these regulatory requirements, there's also been an important working group, the PQRI, or Product Quality Research Institute, that have been publishing recommendations for extractables and leachables testing. So in 2006, they published a recommendation for orally inhaled and nasal drug products. And in the last years, they have also been working on recommendations for parenteral applications. And it is expected that those recommendations will be published this year. And these recommendations will also contain threshold levels for the assessment of extractables and leachables, which are quite important in the evaluation of these studies. So these past guidelines that we discussed are all focusing on drug products, but beside drug products, there are also medical devices that are solutions that are stored in a large volume container and are thus quite similar to large volume parenteral drug products. Because these are medical devices, they fall under a different guideline, the ISO 10993, and they are categorized in this guideline as externally communicating devices. The ISO 10993 contains a chapter 18, which deals on chemical characterization of those medical devices. And in that chapter, it is mentioned that for those devices, one can focus on the leachables assessment. The guideline also contains a table which gives recommended extraction conditions according to the contact category of the medical device. And although that some of those medical devices, infusion bags, might be administered maybe only once to a patient over a short period of time, the solution is in contact with the bag system over a longer period of time or a longer shelf life, and therefore one should take into account that the extraction conditions that need to be applied might be more appropriate to be in line with long-term contact devices. So let's look now at what are the sources of extractables and leachables in flexible bag systems. So the primary container or the flexible bag consists of different components. And the major component is the primary packaging film, which is usually a multi-laminated film, and the internal contact layer often consists of a polyolefin, but it can also consist of other materials. So there is quite a wide variety of films available on the market, and the extractables profile will of course largely depend on the material that is selected. So the primary packaging film has the largest contact surface area with the solution that is stored in the bag, and will thus obviously have a large impact on the extractables and leachables profile. But besides the primary packaging film, there are also ports attached typically to a flexible bag, and that ports may contain tubes, twist-off ports, stoppers, and so on. And those materials have minimal contact with the solution stored inside the bag, but still they can have a high impact on the leachables or extractables profile. So it's important to also, for these materials, select materials that are high in quality. One should also take into account that some polymers, such as polyisoprene, for instance, that is sometimes used as a stopper in flexible bag systems, can contain solvents such as dibenzylamine and dibutylamine, which might be precursors for nitrosamines. On the outside of the primary bag system, usually a label or printing is attached, and due to the semi-permeable character of the primary film, compounds of these labels and inks might also migrate into the contact solution through the primary packaging system. So typical compounds of labels and inks can also be detected in the contact solution. A bag system is usually packed in an overpouch to protect it from the environment, from light or from oxygen or evaporation. And so this overwrap can also have an important impact on the extractables and leachables profile. The overwrap is usually also a multi-layer film. There might be aluminium present in this film to protect the content from light. And often polyurethane adhesives are used to keep the different layers together. And these polyurethane adhesives can introduce typical extractables or leachables, which are bislactone compounds and cyclic esters. So important to remember is that even the secondary packaging materials and the packaging materials that have minimal contact with the solution stored in the bag system might still have a high impact on the leachables profile due to the semi-permeability of the primary container, high solubility of more polar migration compounds in the typical aqueous infusion solutions, such as those cyclic esters related to polyurethane adhesives. The secondary packaging system also creates a closed environment which traps the extractables and makes them more easily migrate into the contact solution. And for a lot of infusion solutions, thermal sterilization by steam sterilization is applied. This goes hand in hand with high temperature conditions, so quite harsh conditions that makes compounds from the packaging system migrate more easily into the contact solution. So this slide shows a case study on the importance of the assessment of secondary packaging. So it's a sodium chloride solution that is stored in a polypropylene bag and that is packed in an aluminium overpouch and was sterilized by steam sterilization. And you see in the GCMS chromatogram the peaks with the asterisk are the highest peaks in the chromatogram, and those are all linked to cyclic esters attributable to the polyurethane adhesive layers of the aluminium overpouch. So let's look then at the challenges for these type of applications. So large volume parenteral application comes with a high dose, which results in challenging analytical and toxicological evaluation. So the authorities are also well aware of that and will look at large volume parenteral applications with high concern. So it's mainly this high dose that causes the challenge. And that originates in the threshold levels that are used for the assessment of extractables and leachables. And as I mentioned earlier, the PQRI has been working on a recommendation for assessment of extractables and leachables, which contains threshold levels for the evaluation of these compounds. And they launched the concept of the safety concern threshold. And the safety concern threshold is a threshold below which a leachable would have a dose so low as to present negligible safety concerns from carcinogenic or non-carcinogenic toxic effects. And the safety concern threshold may vary depending on the route of administration. And so for parenteral drug products, the PQRI will be proposing a threshold level of 1.5 micrograms per day for mutagens and 5 micrograms per day for sensitizers and compounds with other toxicity effects. These safety concern thresholds are expressed in micrograms per day, but the results of an analytical study on extractables and leachables are mostly expressed in other units, such as microgram per unit, for instance. So the ACT needs to be divided by the maximum administered units per day in order to calculate an analytical evaluation threshold, or AET. In addition, screening methods are used for the evaluation of extractables in an extractable study, and the results are considered estimated or semi-quantitative. And this comes with an analytical uncertainty factor that also needs to be taken into account when setting the AET level. And the cornerstone of ANL testing is that all the compounds that fall below this final AET do not need further toxicological assessment or identification. So as we mentioned, large volume parenterals come with a high dose of at least 100 milliliter or more, and this will thus result in a low AET because in the AET you are dividing by the dose. So we can illustrate that with an example. If you have a bag system that is filled with a sodium citrate solution, for instance, that has a filling volume of 250 milliliters and the maximum administered doses per day is 2 bags, then we can calculate the AET by dividing the threshold level with that maximum administered dose. So this results in an AET of 0.75 micrograms per bag for the mutagens and 2.5 micrograms per bag for sensitizers and irritants. And if we convert that in microgram per liter, so microgram per volume, this corresponds to 3 micrograms per liter and 10 micrograms per liter. So those are really low analytical levels, and due to these low analytical levels, more compounds will be detected and reported, and more compounds will need to be identified. So there's more analytical work to be done. Besides the threshold levels as being put forward by the PQRI, probably most of you are also familiar with the ICH-M7 guideline on mutagenic impurities. And the ICH-M7 guideline launches the concept of TTC, threshold of toxicological concern. And this TTC there's a stage TTC proposed that is based on the therapy duration. So the longer the duration of the therapy, the lower the TTC, or the shorter the therapy duration, the higher the TTC. It should, however, be clear that the ICH-M7 guideline is only applicable for mutagenic impurities, and it relates to an excess cancer risk of 1 in 100,000 over a lifetime exposure. So a stage TTC can be applied for mutagenic impurities based on ICH-M7. However, as we saw in the PQRI thresholds, also sensitizers and irritants need to be assessed in the extractables and leachables studies, and those compounds for those compounds, a staged approach is not possible since they have an immediate toxic effect for the patient. So for a study, it will always be needed that compounds are evaluated at least at a 5 micrograms per day threshold in order to take into account this potential irritation or sensitization effect. Then finally, we will look at the different setups that can be used for extractables testing, material assessment versus packaging assessment. So this slide shows the safety assessment triad that was introduced by our expert Dennis Jenke. And that's a three-step approach that is ideally followed to set up extractables and leachables assessment of a packaging system. So it starts with a material assessment where the packaging components are exposed to a controlled extraction study and that generates worst-case extractables data. The second step is a packaging assessment where the packaging system as a whole is subjected to a worst-case simulation study and that enables to identify extractables as probable leachables. And then finally, there is the product assessment or the leachables study that analyzes the confirmed leachables. So in this presentation, we will focus on the first two steps, which are considered as extractables testing. So let's look first at the material assessment. So in the material assessment, the different components of the packaging system are assessed separately. So they should be subjected to sterilization processes prior to the extractable study. And they are extracted under worst case conditions. So that's, for instance, closed vessel extraction or reflux extraction. And the extraction solvents that are selected should also be worst case for the final application, or if multiple applications needs to be covered, generic solvents can be used. So typical solvents can be water at a low pH, water at a high pH, and organic solvents. So the advantages of this approach of doing a material assessment is that it's possible to track the source of the leachables later on to the component of the packaging system that is introducing those. By submerging the components in the extraction solvent, one can also achieve lower reporting limits by playing with the extraction ratio, and it will be more easy to reject those components, those materials that cause safety concerns. The downside is that by extracting all these components separately under more harsh conditions, one generates a long list of extractables for each material separately, and when this information is all added up, you end up with a very long list of compounds, and it will become more difficult to select the relevant targets to be further followed up in a leachable study. So the purpose of the material assessment is mainly to chemically characterize the candidate materials, to identify as many compounds as possible that can migrate from the materials, and to identify bad actors in order to reject them in time. The focus of this study is on identification, so screening analyses are used. Preferably a combination of different analytical techniques is used that has the purpose to discover and identify a wide range of compounds. At Nelson Labs, we also have a proprietary screening database available for identification of compounds that contains more than 5,000 compounds. So that facilitates, of course, the identification. And my colleague Huerto Tri will talk further on that in his presentation. So let's look at a case study of such material assessment. So we have a bag system that consists of a primary packaging film that is printed, a primary packaging tube and a twist-off port, and that is packed in an aluminum overpouch. The solution that is stored inside this bag system is an antibiotic with a pH of 4.5. The extraction of the different components, different materials, was done by closed vessel extraction and neat headspace enrichment by cutting the film in pieces, submerging it into the extraction solvent, or putting it in a headspace vial and heat it, while the tube and the twist-off port were extracted as such. The extraction solvents that were selected were 10% isopropanol in ultra-pure water, which is a worst case for the organic content of the drug product, and that was exposed at conditions at 10 degrees below the boiling point for 24 hours. In addition, an extraction in ultra-pure water was performed, adjusted to pH 4.5 to mimic the aqueous character of the drug product and its pH. And the conditions for extraction there were autoclavation at 121 degrees for 30 minutes. So to calculate an AET for this study, a maximum daily dose of 1200 milliliter was used, or 12 bags of 100 milliliter. And this product was also available in different filling volumes. And in order to calculate an AET, the worst case filling volume was selected. In most cases, this will be the lowest filling volume because the bag with the lowest filling volume typically has the highest surface area versus filling volume ratio. So a worst case amount of surface area for film is used, and the worst case number of ports is used to calculate the AET for this study. And in this way, the AET and the results of this study can be expressed in units per surface area or units per number of test items, and it makes it more easy to extrapolate the results to the other filling volumes. So the calculation of the AET is listed here in the table. So the SET is divided by this worst case value for each component separately. So you end up with an AET for each packaging component separately. If we then look at the results, this table shows the number of compounds that were detected over the different techniques for the different packaging components, and you end up with a total of 382 compounds. I think it's obvious that it will not be possible to follow up 382 compounds in a leachable study. So there will be a need to narrow down this number, and even only with toxicology, this will be difficult because also for the toxicologist, it will be a tremendous task to perform toxicological assessment for such a high number of compounds. So that's where the packaging assessment comes in. So the packaging assessment is an assessment on the packaging system as a whole, and it is subjected to simulating conditions. So the extraction conditions typically are filling and incubation of the packaging system and then subjecting it to accelerated aging conditions. The extraction solvents are also preferably chosen to simulate the final application or bracket different final applications. And if it's possible, the drug product vehicle can also be used for extraction. So the advantages of this approach is that the extractables profile generated will be much more close to reality, and it enables you to narrow down the list of potential leachables. If you set up this packaging assessment well, different applications can be bracketed, and because you are using clean solvents instead of the drug matrix, such as what is being analyzed in a leachable study, it's still possible to achieve lower reporting limits. The downside of this approach is that the source of the extractables is unknown. So one should be well aware of that, because we see sometimes that clients go immediately to a packaging assessment and skip the material assessment to prepare for the leachable study. But the downside is if you detect an extractable compound of concern, it will be difficult to track down the source and reject that component of material that is introducing that compound of concern. So the purpose of a packaging assessment is to establish a worst-case accumulation of potential leachables and identify those compounds that need to be monitored in the leachable study. So it really builds a bridge between the material assessment and the leachable study. And again, preferably screening analysis is used for this assessment. But still, you should keep in mind that a toxicological assessment might still be needed to evaluate the data of the simulation study in order to limit the design of the leachable study. So let's look again at the case study for a packaging assessment. So we have the same product, the same bag system, and extraction is done by filling and incubation with 6% isopropanol in ultra-pure water at pH 4.5. The aging conditions used were accelerated according to the ICH guideline on stability studies, so it was aged for 6 months at 40 degrees. An alternative for accelerated aging might be to follow the ASTM standard for accelerated aging of medical devices. A safety concern threshold was calculated in the contact solution by taking into account the maximum daily administered dose to the patient of 1200 milliliter. And because the product was actually non-chronically dosed, the safety concern threshold of 5 micrograms per day was used as a starting point. And in that way, an AET level of 4.2 micrograms per liter was reached. So if we look then at the components or the compounds that are detected in that study, only 56 compounds were detected in total. So that's already a tremendous reduction versus the material assessment, and it will be more easy for the toxicologist to assess these 56 compounds and select relevant leachables from that set. So in conclusion, an extractable study on a flexible bag system should include all components of a packaging system, including also components with only minimal direct product contact and secondary packaging. The high dosing regimens of a large volume parenteral application result in low analytical evaluation threshold, and that results in higher analytical and toxicological workload. And ideally, a 3-step approach is used for evaluation of large volume parenteral applications, consisting of a material assessment, a packaging assessment, and then a product assessment. So this brings us to the end of the presentation. I thank you for your attention. And if you have any questions, we welcome them, of course, and they can be further discussed in the panel discussion or in a one-on-one discussion. So thank you for your attention.